Today we are going to discuss about the topic glycolysis, also known as the Enden-Meyerhof pathway. Glycolysis takes a central part in metabolism. It is a breakdown of a sugar molecule. Now we all know that we require energy for the functioning of our body, and the main source of energy in our body is glucose. Now this glucose can come from anywhere. It might come from our diet, or it might come from breakdown of glycogen, which is a storage product of carbohydrate, or it may be synthesized in the body, for example, through gluconeogenesis. But ultimately, the glucose molecule has to be broken down to give us energy. And this process of breaking down glucose is called glycolysis. So what is glycolysis? It comes from the Greek word glykis, meaning sweet, and lysis, meaning splitting. So it is the splitting of a glucose molecule. Chemically, it consists of a cytoplasmic reaction sequence of 10 enzyme catalyzed reactions producing 2 molecules of pyruvate from glucose. So we start from 1 molecule of glucose and we produce 2 molecules of pyruvate. So we start from 1 6-carbon compound, that is glucose, and 2 3-carbon molecules, that is 2 molecules of pyruvate. The whole process is an anaerobic process, and it was first found out in yeast by Warburg and Chaplin and muscle by Enden and Meyerhof. Hence, we also call it the Enden-Meyerhof pathway. Now, why is glucose utilized as a source of energy? It is because glucose has a very high potential energy. On complete oxidation of glucose to carbon dioxide and water, the free energy released is 2840 kilojoules per mole. This is a very good source of energy. What is the role of glycolysis? We know that the primary role is production of energy, and glucose itself is the sole source of energy for many organs and cells, like also the RBCs, the renal medulla, the brain, sperm, and certain other cells also. Other than this, it provides intermediates for other metabolic pathways. So as we know, glycolysis is a sequence of 10 metabolic reactions, and intermediates are produced, and these intermediates might be used in other metabolic pathways as well. Coming to the reactions of glycolysis, broadly the reactions are divided into 2 phases. Number 1 is the investment phase, and number 2 is the payoff phase. The investment phase consists of the first 5 reactions. Here glucose is activated and ultimately energy is utilized here. So that's why we call it the investment phase. In the payoff phase, it is the phase where it is the second 5 reactions, and it is this phase where energy is produced in the form of ATP. Now these are the steps of glycolysis. I won't be discussing them now because I'll be discussing the reactions one by one after this. Before we go into the reactions, I'd just like to have a brief revision of the glucose structure. I believe that knowing the structure of glucose is very important, because if you understand the numbering of the glucose, you will remember the substrates and the products and also the enzymes which are involved. So we know that glucose is a 6-carbon compound. This is the ring representation of the glucose structure. Okay, and the carbons, it is an aldol sugar, and the carbons are numbered, carbon number 1, 2, 3, 4, 5, and 6. So when we talk of glucose 6-phosphate, we mean that a phosphate group has been attached to the glucose at position number 6. Now coming to the reactions of glycolysis. Step 1, phosphorylation of glucose. It is a very simple but a very important reaction. Here glucose is phosphorylated at position number 6. So this is position number 6. A phosphate group is added here. So from glucose we are producing glucose 6-phosphate. The enzyme involved here is hexokinase. It can also be glucokinase. And here the phosphate groups comes from ATP. So we are utilizing 1 molecule of ATP in this reaction. This negative charge of the phosphate, it prevents the glucose 6-phosphate from leaving the cell, so it becomes committed to glycolysis. 1 ATP is utilized, and this reaction can be catalyzed either by hexokinase and glucokinase. So next the question comes that where is hexokinase used and where is glucokinase used? And very important, this is an irreversible reaction, so this is one of the points which is subject to regulation. Now the difference between hexokinase and glucokinase. Both of them are kinase, both of them can phosphorylate glucose. But what is the difference between them? See, the function of hexokinase, it is the main enzyme of glycolysis, whereas glucokinase, it performs the same function but only when the levels of sugar are very high, for example in the postprandial state. So, hexokinase, it is active in most tissues, whereas glucokinase is active only in the liver. Hexokinase can act on any hexose molecule, whereas glucokinase is specific for glucose. The Km value of hexokinase is low, indicating that the affinity is high and it can act on glucose even when the concentration of glucose is very low. Opposite happens in case of glucokinase. It has very high Km value and a low affinity, or in other words, glucokinase will attack glucose, react on glucose only when the concentrations are very high. This goes along with its function that glucokinase comes into action only when the levels of glucose are very high. The Bmax is low in case of hexokinase and high in case of glucokinase. Hexokinase is subject to feedback inhibition by glucose 6-phosphate, whereas glucokinase is not. Now, this is also very relevant because glucokinase only functions when the levels of glucose are high. So, its function is to bring down the levels of glucose, and we do not want it to be inhibited by glucose 6-phosphate. Next reaction is isomerization of glucose 6-phosphate to fructose 6-phosphate. It is a very simple reaction consisting of the conversion of glucose to 6-phosphate to fructose 6-phosphate, that is the aldose sugar, to its isomer, which is a ketose sugar. So the enzyme here is an isomerase and it is a reversible reaction. Step number 3, phosphorylation of fructose 6-phosphate. This is a very, very important reaction. First of all, the enzyme involved here is phosphofructokinase 1, which we commonly call PFK1. This is a major point of regulation of glycolysis. It is the committed and the rate limiting step, and it is irreversible. So you see here that a phosphate group is attached at position number 1. So here we have the phosphate group attached. We already have the phosphate at position number 6. So the product here is fructose 1,6-bisphosphate. We call it bisphosphate because the two phosphate groups are attached at two different positions. If both were attached to the same position, we would have called it biphosphate. The phosphate group here comes from ATP, so we are utilizing another molecule of ATP in this reaction. Next is the cleavage of fructose 1,6-bisphosphate. So it is this fructose 1,6-bisphosphate is simply sliced in the middle with the help of the enzyme aldolase, giving dihydroxyacetone phosphate and glyceraldehyde 3-phosphate. So here is where we are converting the 6-carbon compound to 2 3-carbon molecules. Next is isomerization of dihydroxyacetone phosphate. Now for the further reactions which take place, only glyceraldehyde 3-phosphate can serve as a substrate. So whatever dihydroxyacetone phosphate was produced, it has to be isomerized into glyceraldehyde 3-phosphate. Now this reaction is the final reaction of the investment phase. It is very important to note here that until now we were dealing with only one molecule, but now one molecule of glucose has been broken down into 2 molecules of glyceraldehyde 3-phosphate. So all the reactions which happen subsequently have happened twice. So when we are calculating the energetics of the reaction, we have to consider it into 2. Every step has to be considered twice. A brief recap of the investment phase. We start with glucose, which is converted to glucose 6-phosphate by the enzyme hexokinase or glucokinase. It is then isomerized to fructose 6-phosphate. Then PFK1 comes into action and converts it to fructose 1,6-bisphosphate. Now this molecule is sliced into 2 and converted to glyceraldehyde 3-phosphate and dihydroxyacetone phosphate. This is the investment phase, and we are utilizing 2 molecules of ATP over here, one during the conversion of glucose to glucose 6-phosphate, and the second during the conversion of fructose 6-phosphate to fructose 1,6-bisphosphate. Next, we come to the payoff phase. So the first reaction here is the dehydrogenation of glyceraldehyde 3-phosphate. Very, very important reaction. The enzyme here is glyceraldehyde 3-phosphate dehydrogenase. So you can see glyceraldehyde 3-phosphate is dehydrogenated. A phosphate group is added, and what is formed is 1,3-bisphosphoglycerate. Now it is very important to note here that though we are adding a phosphate group, the source of this phosphate is not ATP, but in else it is inorganic phosphate. So we are not utilizing ATP over here. Second, this is accompanied by a dehydrogenation reaction. So NAD is converted to NADH and H+, and we all know very well that what happens to NADH, it enters into the electron transport chain, and from each molecule of NADH, we are getting 2.5 moles of ATP. So the energy production here is 2.5 into 2, that is 5 ATPs are produced from this reaction. Next, dephosphorylation of 1,3-bisphosphoglycerate. So here we have a phosphate group which is removed, and the receiver of the phosphate is ATP. So we are getting one molecule of ATP. Now, very important that this reaction, it is an example of substrate level phosphorylation. That is, ATP is being produced directly without the help of the electron transport chain. So this reaction is an example of substrate level phosphorylation. One ATP is being produced. The reaction is taking place twice. So 2 ATPs is the energy production from this step. Next is the shift of the phosphate group. It is catalyzed by the enzyme phosphoglycerate mutase. So the phosphate is shifted from position number 3 to position number 2. Reaction number 9. Dehydrogenation of 2-phosphoglycerate. 2-phosphoglycerate is dehydrated, and the enzyme here is enolase. Again, this is a reversible reaction, and what we are getting is phosphenol pyruvate. So there is this redistribution of energy within the same molecule, leading to the formation of a very energy-rich compound capable of forming ATP. Final reaction is the formation of pyruvate. So phosphenol pyruvate, the phosphate is removed here and it is given to ADP, and what we are getting here is one molecule of ATP. So this again is an example of substrate level phosphorylation, and here one ATP is being produced. The reaction is taking place twice, so 2 ATPs are getting produced from this step. So phosphenol pyruvate along with ATP in the presence of the enzyme pyruvate kinase, we get pyruvate and one molecule of ATP. And this is the third irreversible reaction of glycolysis. An applied part of glycolysis that in case of pyruvate kinase deficiency, this is a type of hemolytic anemia. So if pyruvate kinase is deficient in the RBCs, what happens that the ATP generated is insufficient for the functioning of the sodium-potassium pump. So the ionic gradient across the cell membrane cannot be maintained, and this leads to cell lysis. So actually we have a pyruvate kinase deficiency, which is an enzyme of glycolysis. If glycolysis is interfered with, then the amount of ATP being produced is insufficient for the proper functioning of the sodium-potassium pump. Since the ionic gradient cannot be maintained, it leads to the increased osmotic pressure and cell lysis. So it is one of the causes of hemolytic anemia. Now we can just have a brief recap of the steps of glycolysis before we go into the energetics and regulation. So first we have glucose, then glucose 6-phosphate, next we have fructose 6-phosphate, fructose 1,6-bisphosphate, which is sliced into glyceraldehyde 3-phosphate and dihydroxyacetone phosphate. Then we have an isomerization reaction take place. Next, glyceraldehyde 3-phosphate is converted to 1,3-bisphosphoglycerate. A phosphate group is removed, giving 3-phosphoglycerate. Positioning of the phosphate is changed, giving 2-phosphoglycerate. Then we have phosphenol pyruvate, and finally the last substrate level phosphorylation, which is pyruvate. Now we have come to the end of the glycolysis, and pyruvate is produced. Now what happens to this pyruvate depends on whether the condition is aerobic or anaerobic. When we are talking about an anaerobic condition, that what happens, that pyruvate in the presence of the enzyme lactate dehydrogenase is converted to lactate. Whereas if it is an aerobic condition, then pyruvate it is converted to acetyl-CoA, and with the help of the enzyme pyruvate dehydrogenase, and acetyl-CoA then enters the TCA cycle. Okay, so here we have glucose through glycolysis giving 2 molecules of pyruvate. In aerobic conditions, It is converted to two molecules of acetyl-CoA, which enters the TCA cycle. And in anaerobic conditions, pyruvate is converted to lactate with the help of the enzyme lactate dehydrogenase. Now this reaction, converting pyruvate to lactate, this is done with the help of the enzyme lactate dehydrogenase. So this happens only in anaerobic glycolysis. The enzyme here is lactate dehydrogenase, and what is very important here is that NADH is being used as the coenzyme and it is being converted to NAD+. So what is the importance of this? That whatever NADH was being produced in glycolysis during the conversion of glyceraldehyde 3-phosphate to 1,3-base phosphoglycerate, that it is being utilized over here. So the energy generated from that step is nil. Okay. So the clinical significance is that it regenerates NAD plus for step number 6, that is dehydrogenation of glyceraldehyde 3-phosphate. At the same time, since the NADH is used up, the NADH cannot enter into the electrotransport chain. Now, this anaerobic glycolysis, it occurs in tissues lacking mitochondria, like in the RBCs, the lens, the cornea, and the renal medulla. In oxygen deficient tissues, for example, severely exercising muscles where the rate of glycolysis is very high, more pyruvate is produced and the oxygen supply is still less due to muscle contraction. So pyruvate is converted to lactate. So initially the glycolysis here is aerobic, but since the muscles are contracting fast and the blood supply is also being compromised due to the muscle contraction, the oxygen supply is insufficient and then they shift to anaerobic glycolysis. In hypoxia due to vascular obstruction, like in acute MI, it helps the cells to survive a brief anoxic episode. So though oxygen is not there, ATP is still produced with the help of anaerobic glycolysis, but that is only for a brief period of time. And these carbohydrates are the only metabolic substrates which are capable of producing ATP anaerobically. Now, the energetics, a brief revision, we've studied it, we've gone through it while we were studying it, but now just once more. During the formation of glucose 6-phosphate from glucose, 1 ATP is utilized. Fructose 6-phosphate to fructose 1,6-bisphosphate, another ATP is utilized. Glyceraldehyde 3-phosphate to 1,3-bisphosphoglyceride, 2.5 into 2, that is 5 ATPs are being produced because NADH is being produced here, which enters the electron transport chain. Conversion of 1,3-base phosphoglycerate to 3-base phosphoglycerate. This is an example of substrate level phosphorylation. So 2 ATPs are being produced here. Another substrate level phosphorylation is conversion of phosphanol pyruvate to pyruvate. 2 ATPs are again being produced here. So totally 9 ATPs are being produced and 2 ATPs are being used up in the investment phase. So finally we have a net amount that is 7 ATPs are being produced per molecule of glucose which is lysed through glycolysis anaerobically. In case of anaerobic state, the ATP which is being produced through the electron transport chain is absent because the NADH is being used up. So here the net ATP production will be 2 ATPs. So in aerobic state we have 7 ATPs which are being produced, whereas in case of the anaerobic state only 2 ATPs are being produced. Now, regarding anaerobic metabolism, I'd like to talk about one more effect, which we also call the Pasteur effect or the Warburg effect. Now, this is a regulatory phenomenon of carbohydrate metabolism, and it has been found that under anaerobic conditions, the tissues utilize much more of glucose than in the aerobic conditions. Or, in other words, there is an inhibition of glucose glycolysis in the presence of oxygen. Now, this can be explained physiologically also because as we already saw that the energy production during aerobic glycolysis is 7 molecules of ATP from 1 glucose molecule, whereas in anaerobic conditions it is only 2 ATPs produced from 1 glucose molecule. So, to satisfy the needs of energy requirement, it is necessary to break down more molecules of glucose under anaerobic conditions. So, there is an inhibition of glycolysis by the presence of oxygen. We also call it the Warburg effect or the Pasteur effect, and it is due to the inhibitory effect of citrate and ATP, which are produced only in aerobic conditions. And we will see in the next slides that how citrate and ATP both act as allosteric inhibitors of glycolysis. Now coming to the regulation of glycolysis. Regulation of glycolysis is very, very important. So we'll be going into it into a little bit of detail. First of all, I would like to stress 2 points. Number one, when we talk about the rate of glycolysis always depends on the overall energy content of the cell. Now, when we are talking about the energy content of the cell, what do we mean? We mean the amount of energy in the terms of ATP. Now, the purpose of glycolysis is to break down glucose and give us ATP. So, obviously, if the energy level is high or we have sufficient ATP, then glycolysis can be inhibited. So, what are the indicators of high energy levels of the cell? ATP, citrate, these indicate a high energy level of the cell. And what about low energy? AMP, ADP, these indicate a lower energy level of the cell. So if we have higher concentrations of AMP and ADP, then glycolysis is stimulated. Number two, the three irreversible steps of glycolysis are subject to regulation. So out of the 10 steps, only step number 1, step number 3, and step number 10 are reversible. So these are the 3 steps in which regulation can take place. Now we will go through these steps one by one. Step number 1, the conversion of glucose to glucose 6-phosphate in the presence of hexokinase. It is subject to feedback inhibition by glucose 6-phosphate. So when the levels of glucose 6-phosphate become high, then hexokinase is inhibited. Remember that I always told you that this step can also be catalyzed by glucokinase. But glucokinase is not subject to feedback inhibition by glucose 6-phosphate because glucokinase functions only when the levels of glucose are high, and the purpose is to bring down the levels of glucose. So it is not subject to feedback inhibition. Number 2, step number 3, the conversion of fructose 6-phosphate to fructose 1,6-bisphosphate catalyzed by PFK1. This can be subject to both allosteric and covalent regulation, and we will be discussing it in detail later. Finally, step number 10, the conversion of phosphanol pyruvate to pyruvate by the enzyme pyruvate kinase. This again is subject both to allosteric and covalent regulation. Now coming to PFK1, phosphofructokinase. It is the most important control point for glycolysis. It is subject to allosteric regulation and also to covalent modification. So what are the allosteric activators? As we already told, that when the energy levels of the cell are low, then glycolysis is activated. So we have AMP and ADP. Other than this, we have potassium phosphate and very important fructose 2,6-bisphosphate, which is a major allosteric activator of phosphofructokinase. How it is going to function, we will be dealing with it in the next slide. And the inhibitors are ATP, citrate, calcium, magnesium, and low pH values for acidic conditions. Now coming to fructose 2,6-bisphosphate, which we told is an allosteric activator of PFK1. Now where does this fructose 2,6-bisphosphate come from? Fructose 2,6-bisphosphate is the most important allosteric activator of PFK1. It is synthesized from fructose 6-phosphate by an enzyme called PFK2, and it is hydrolyzed back to fructose 6-phosphate by fructose 2,6-bisphosphatase 2. Now both these PFK2 and fructose 2,6-bisphosphatase 2 are catalyzed by the same polypeptide, which is therefore a bifunctional enzyme. Now if we look at it again, fructose 6-phosphate, it is a normal intermediate of glycolysis. It is converted, during glycolysis, it is converted to fructose 1,6-bisphosphate. Other than this, there is an alternative pathway in which fructose 6-phosphate is converted to fructose 2,6-bisphosphate. So if we can see the difference between the two, there is only a difference in the position of the phosphate group. Now the first initial reaction is catalyzed by PFK1, and the second reaction is it is catalyzed by PFK2. Now the importance of this fructose 2,6-bisphosphate is that it has an upregulatory effect on PFK1. That is, it stimulates PFK1, or in other words, it will increase the rate of glycolysis. If the fructose 2,6 bisphosphate is not required or glycolysis has to be inhibited, then fructose 2,6 bisphosphate, it is hydrolyzed back to fructose 6-phosphate, and the enzyme here is phosphatase. Now, very important that this PFK2 and phosphatase both are parts of a single enzyme, in other words, a bifunctional enzyme. So, if we have a brief revision, we can start with fructose 6-phosphate, which is normally converted by PFK1 to fructose 1,6 bisphosphate. Alternative product is fructose 2,6 bisphosphate. The enzyme in case of the first reaction is PFK1, and here it is PFK2. Now, this fructose 2,6 bisphosphate upregulates PFK1, and it can be hydrolyzed back to fructose 6-phosphate by the enzyme phosphatase. And this PFK2 and this bisphosphatase, they are both a part of a single bifunctional enzyme. So we have fructose 6-phosphate converted to fructose 2,6-phosphate bisphosphate by PFK2 and hydrolyzed back by fructose 2,6 bisphosphatase. Now, the balance between the two depends on the levels of insulin and glucagon, which we will be coming to later. Now, coming to the covalent modification of PFK2. Now, we have seen that here in this slide that PFK2, it is a bifunctional enzyme. It can act as a kinase or a phosphatase. So, what determines whether the enzyme will act as a kinase or it will act as a phosphatase? Now, this in turn depends upon the covalent modification of the enzyme. Now, this PFK2, it can exist in two states: phosphorylated state, dephosphorylated state. In the phosphorylated state, the PFK2 acts as a phosphatase, and dephosphorylated state, it will act as a kinase. So, go through it once again. PFK2, two states: phosphorylated, dephosphorylated. Phosphorylated state acts as a phosphatase. Dephosphorylated state acts as a kinase. So, here we have fructose 6-phosphate to fructose 2,6 bisphosphate. This is catalyzed by the dephosphorylated form, which acts as the kinase, and the hydrolysis is done by the phosphorylated form, which acts as a phosphatase. Now, what will determine which way the bifunctional enzyme will act? Will it act as a kinase or will it act as a phosphatase? So, both of them will have opposite effects. Now, which way the bifunctional enzyme will act at a particular point, that is determined by the levels of insulin and glucagon. Now, if you think over it, what is the function of insulin and what is the function of glucagon? Both regulate the levels of glucose in the blood. Glucagon is secreted when the levels of sugar are low. So we have to bring up the glucose levels. And how is the glucose levels brought up? Decreasing glycolysis. And insulin, in turn, it increases the entry of the sugar into the cells, increases the rate of glycolysis, and the blood glucose comes down. Glucagon will be inhibiting glycolysis and it will be stimulating gluconeogenesis. So remember the end point of each hormone to remember the mechanism. Now, when we talk about the mechanism of action of glucagon, now glucagon and insulin, both of these act through second messengers and cyclic AMP dependent protein kinase mediated pathway. So glucagon, when the sugar levels are low, glucagon is secreted from the pancreas, and glucagon travels through the blood and reaches the target cells. Now, what does glucagon do? It increases through the action of second messengers. It increases the levels of cyclic AMP, increases the activity of cyclic AMP dependent protein kinase. This phosphorylates PFK2, and in the phosphorylated state, the PFK2 acts as a phosphatase. So the levels of fructose 2,6 bisphosphate are decreased. So this decreases the activity of PFK1, decreases glycolysis, and blood sugar levels will increase. So we have a revision. Glucagon increases cyclic AMP, increases the activity of cyclic AMP dependent kinase, phosphorylates PFK2, stimulates the phosphatase activity, decrease levels of fructose 2,6 bisphosphate, decrease PFK1 activity, decrease glycolysis, and obviously as an output, increase in the levels of glucose. Coming to insulin, opposite effect. Insulin decreases cyclic AMP, decreases the activity of cyclic AMP dependent kinase, dephosphorylates PFK2, so it acts as a kinase. This leads to the increased levels of fructose 2,6 bisphosphate, increased activity of PFK1, increased glycolysis, and more movement of the glucose inside. So more glycolysis is stimulated. Now coming to the regulation of pyruvate kinase. Pyruvate kinase similarly is subject to regulation by allosteric regulation and covalent modification. Allosteric activation, it is activated by fructose 1,6 bisphosphate and it is inhibited by alanine and ATP. Covalent modification, similarly it is inhibited by phosphorylation through glucagon. Finally, we come to the inhibitors of glycolysis. Fluoride, it inhibits enolase, which catalyzes step number 9 of glycolysis. This is very important because fluoride, it is commonly used along with potassium oxalate in blood, which is collected for blood glucose estimation. So when we collect blood, a patient's blood for estimation of glucose, which is a very important and practical test, what happens? We collect the glucose with the blood, but do we estimate the glucose immediately? No. There is some time lag between the time in which we are collecting the blood and the time when the blood sample is being tested. If glycolysis is not inhibited, glycolysis will continue and the levels of glucose will fall. So glycolysis has to be inhibited, and this is inhibited by adding fluoride along with potassium oxalate in the samples which are collected for glucose estimation. Iodoacetate, which inhibits glyceraldehyde 3-phosphate dehydrogenase, and arsenite, which inhibits phosphoglycerate kinase. So thank you very much. That is the end of the presentation. And if any of you have any doubts or any queries, you are free to contact me. Thank you.